Thank you very much, David. I'm going to talk today about some of our work on reconstructing regulatory circuits. And I'm going to start with yeasts and tell you a little bit about work that we've done several years ago, because that formed the foundation in the way that we think about these problems computationally. And then in the second half of the talk, I'm going to show you how we're taking the approaches that we've developed in yeast, both computationally and in terms of experimental strategies, and integrate them together in order to resolve the much more complex regulatory circuits that exist in mammals. So first of all, what are regulatory circuits? Well, regulatory circuits are the circuits that control the RNA levels that exist in cells. They take two types of inputs. They take inputs that we call trans inputs, and these could be things like transcription factors that bind to DNA sequences, or other types of molecules, including small molecules in the environment. They integrate them with cis information, which is encoded in the DNA sequences, either proximal or further away from the body of a gene. And from this...
What's cis?
Cis means that it's in the same physical piece of DNA together with a gene. Whereas trans means that it's not. And you can actually determine these genetically, because if the regulatory impact of a cis element, or the regulatory impact of a cis element is dependent on the fact that it will be attached in the same DNA molecule together with the gene that it regulates. Whereas a trans element, you can encode in a separate piece of DNA and it will still have the same regulatory impact. It's genetics jargon. But it takes these two pieces of information together, which can represent a very large number of molecular elements, and integrates them somehow to determine at a very quantitative and precise level the amounts of RNA of different types of genes in the cell. This RNA will later on get translated into protein. These proteins can get modified. They can get put in different places in the cell. There are many, many additional regulatory layers that in the end control how the cell works. But this is one of the most essential ones, and it is the one on which we focus. Now, with the advent of the genomics, the ability to measure on a very large scale, say, all the different RNA transcripts that exist in the cell, all the proteins that exist in the cell, measuring genome sequences from different individuals and from different organisms, the hope was that we would be able to take these large-scale measurements and reconstruct from them somehow the actual mathematical function or computational function that actually takes these inputs and computes the output out of them. But what mostly happens when you actually take these large-scale data sets and integrate them together is something that looks like this. And this is real data. It's not just made for the slide. Something that looks like this, and obviously it does not necessarily give us tremendous insight. So the fact that you can measure a lot and you can collect a lot does not necessarily mean that you understand a lot about what goes on in cells. And to this day, the vast majority of understanding of the principles that govern gene regulation do not come from large-scale studies. They come from very small-scale studies where people were able to manipulate systems in great details. So what are the principles of these very basic small-scale studies? They consist of 3 steps typically. In the first step, you observe something about the system. Usually you observe what its output is doing. In the second step, you try to model something inferred from these observations. And then in the third step, you will try to perturb different components in the system to observe again the effect of perturbations on the output, from which you can validate or refine your model. Now, this is easier to do when you just have a very small system. There's less to manipulate, there's less to observe, and there's less to model. It's harder to do when you're trying to study this on a genome scale. But never, yes.
Could you explain what these observations are of? Are they at the genetic level or at the phenotype level?
The observations are typically at the molecular level. These are at anything that is not the gene sequence is in fact a phenotype. So a phenotype can be something like the size of the cell or the length of the arm, but it can also be something like the quantity of RNA that exists inside the cell. So we think of those as molecular phenotypes rather than thinking of them as more gross organismal or cellular phenotypes. And the beauty of molecular phenotypes is that we understand a lot about the mechanisms and the molecules that interact together in order to generate them.
So what you observe are molecular
Phenotypes. From them you try to infer what are the molecular mechanisms that lead to those phenotypes. And once you infer a model, you can look at the components of the model, and you can go back experimentally, perturb them again, and see what effects it has on the output.
Is the model at the genotype?
And the model should be at the molecular level as well.
But of genotype, of genes.
Of genes, yeah. Genotype is a slightly different, has a slightly different meaning. Yeah. You will see in a second what a model looks like, and I think it will become clearer then. So nevertheless, when there is the ability to measure, people usually cannot restrain themselves from measuring, so they do that. Here's an example of one kind of measurement technology that we have today. In this case, these are DNA microarrays. They're actually being phased out by now, being subsumed by a different type of technology, but it doesn't really matter for our purposes. This is a technology that lets you measure the level of RNA expressed from every gene in the genome. And the genes in the genome, in this case, this is from the yeast genome, will be here in the rows, so there are approximately 6,000 rows here. The different experiments under which these were measured are going to appear in the columns. These are, in this particular case, experiments of different types of stresses. And these are arrays that actually measure relative levels, in this case, before and after stress. And when the gene's level went down, this will be encoded in this image by a red color. When the gene level when it went up, it will be encoded by a red color. When it went down, it will be encoded by a green color. And the black color will represent no change. And this can be on a nice color scale that will give you nice dynamic range. But this is just for visual representation. And this is from an experiment done several years ago by Audrey Gash in David Botstein's lab when he was still at Stanford.
How many genes are there?
6,000 rows here.
6,000 rows?
Yeah, roughly. 5,900 and something. Yeah. And that's, and a lot of them are not really doing much, right? Their level is not changing in any of these responses that she measured. About 1,000 actually are represented by these big patches of red and green. And you can learn interesting things just from observing the output. For example, you can learn that there's a pretty canonical response to all of these different environmental stresses that they try to apply to the cell. And they coined it the environmental stress response. And it turns out that when cells are stressed out, and it doesn't matter if the temperature goes up, or you put oxidative stress on them with hydrogen peroxide, or you treat them with something that denatures their proteins, or kind of messes their proteins up, or you put high osmotic pressure on them, many different things. There's a whole bunch of genes that the cell will always induce, and there's a whole bunch of genes that the cell will always repress. Okay? These are going to be some big responses, and we're going to look at them.
Is there some vertical ordering here?
These are clustered. So in this particular case, I think they're clustered by hierarchical agglomerative clustering, and the distance is probably correlation, although I'm not 100% certain.
But they were put together.
They were put together in some ordering, and the ordering is using one or the other standard clustering algorithm. And there's actually, it's beyond the cottage industry. There are thousands of papers on how to best cluster gene expression profiles. It's an interesting problem. Actually, one of the side products of one of the methods I'll show today is to give you one of these clusterings. But the bottom line is when data looks nice, it clusters nicely by all methods, and when it's hard to cluster, most methods don't do it well.
So it's clustered by the output or by some other?
It's clustered just based on the output. You know nothing about this system except for the physical thing you did to the cell. Okay, which is hard to translate to molecular mechanism, and by the fact that this is what happened to the output of the system. Yes?
One thing I've heard cell biologists say is that often what we learn from these experiments was in some sense known before, that there was such a response. And then the microarrays may pull out the specific gene cluster. To what extent, for this particular example that you're showing, was the existence of a kind of a core stress response known beforehand, and what was actually learned from this study?
That was actually not as well known before this study. This study is one of the
question. For the
yes. So the question was, and that's actually a very legitimate criticism. The question was, people do these large-scale studies, and then mostly what they find is the known. Because they already knew there was a stress response, and they already measured some, marker genes, 1 or 2 or 10, that reflected it, and they already knew that these genes were changing, and they didn't need the fancy and expensive experiment that measured all of them in order to know what they ended up knowing after doing this big study. That's very true in many cases, but it's actually not true for the environmental stress response. It was not clear because what happened was that in each of these responses, there are also things that are specific, right? You can see here, this is nitrogen depletion. You take the nitrogen away from the cells. There's a response that's very specific to nitrogen depletion and other types of starvation. That's not common across all the stress responses. And most of the output genes that people have looked at during nitrogen depletion had to do with this, because these were the things that made the immediate sense. When you take away nitrogen, you need some enzymes to compensate for the fact that you don't have enough nitrogen around, right? You need to break down some things that you have in the cell to provide yourself with sufficient nitrogen. This was not, so this is what people continuously looked at. Only when they did the array in an unbiased way did they realize that actually there's a much bigger response here, which is part of this shared environmental stress response. Yes?
Why is there a green line on the bottom if it's clustered according to alpha?
Oh, because there's enough other stuff here, right? That these are all grouped together because you have this green together with this green. This is separate because you don't have this green part. And when you do hierarchical agglomerative clustering, if you're familiar with it, the way that hierarchical agglomerative clustering works is that it is actually agnostic in terms of how it arranges the leaves at the bottom of the tree. So if you have two clusters, you could put them like this, or you could put them like this, and it's completely symmetric in terms of the scoring. The result of this is that the image might not look as pretty with one ordering compared to the other ordering. And there's actually algorithms called optimal leaf ordering algorithms that are forced to do perfectly or NP hard, but you can do some nice heuristics. There are such algorithms that actually give you also the best visual. And there's a guy, Ziv Bar Yosef, who works at Carnegie Mellon, that actually has done a lot of work on that. And yes?
In terms of mechanisms,
Yes.
you already assumed that it's only transcriptional factor and cis. There could be other mechanisms. With
I didn't say
microRNA,
it was
could
only
be,
two.
right, could be allelic exclusion, could be isoforms, splicing regulations, so on and so forth. They will not be seen in this model.
First, clarify. I did not say that these are necessarily transcription factors. The cartoon was made for simplicity. I actually said it could be many other molecules as well. In a standard model that we think of, we separate two types of inputs. One input comes along for the ride, and it has to be encoded on the same DNA molecule. That's what we call the cis. The other set of inputs can be anything. It can be your transcription factors, signaling molecules that come upstream of them. It can be large non-coding RNAs. It can be small non-coding RNAs. It can be regulation of the splicing. It can be regulation of the original production of RNA, of the processing of that RNA, of moving that RNA from the nucleus to the cytoplasm, of degrading that RNA, of excluding that RNA in particular bodies called P bodies. Each of these steps is regulated, and when you measure this RNA level here at the end, it's actually the integration across all of these processes and across the time frame at which the cell has been accruing all of these changes. So it's a true integration, like mathematical integration. The reason that people tend to get hung on this part is that their measurement technologies are best for this part. And actually, I don't have it on, but I could pull up these slides if people get curious at the very end of the talk. We have actually worked very recently with new technologies that let you separate each of the independent contribution into this process.
Well, that was my next question. In the measurements, your cells are
Yes.
average, it's time average.
Now you have two problems with these measurements. The first is the time averaging. Actually, when you do these types of things that we tend to denote by these little triangles, you put an impulse on the cell, we call it. It's called an impulse response. You do something to the cell and then it initiates a response. It actually helps you with getting good temporal resolution and distinguishing certain patterns that would be very hard to distinguish just from what we call steady state measurements. Having said that, this is not, it doesn't give you transcription rates. It still gives you integration in time, but at the resolution of the time points that you've been collecting. The other thing that is an issue is the fact that we're measuring across a population of cells. There are still no good genome scale approaches that work at the single cell level. The single cell level has shown, mostly from microscopy studies, that there is tremendous variation between individual cells in the population. That's true when you take these isogenic yeasts. They have exactly the same genome. You grow them from a very small inoculum of cells. Their genome is the same. They're going to express genes differently. They're not all going to express genes in the same way. And what we see here is an averaging of that. They do all sorts of crazy things. For example, they could hedge their bets and decide some of them will respond to the signal, some of them will not. They can do it at different timescales. There could be a lot of differences there. It's also true when you look at in multicellular organisms, when you look at differentiating cells, when you look at cells from cancers. Each cell in a complex cancer can actually have a pretty distinct response inside it, and that is, for example, a big challenge for chemotherapy. So it's absolutely true that this is an averaging. I think in biology, one of the things, it doesn't really matter which choice of which level of resolution or technique that you're choosing. All biologists tend to be very pragmatical. They know that there's a lot that they don't know about the cell that they're measuring. So either they'll try to measure in an unbiased way, but then they might not have as many sample or as many opportunities to perturb or the ability to look at individual cells. Or they might be measuring just one thing, but then they might not have the breadth, or they might be measuring everything. They never get everything that they want, so they try to make the most with what they have. Yes?
Is there a way with this to not, obviously you're losing the temporal dynamics. Some of the most interesting genes I imagine are undergoing oscillations from high to low, rapid autonomous stress. Is there any way of looking at the dynamic range of any of these?
Yeah, so the typical response looked something, I wasn't going to talk about this, but I enjoy talking about this. The typical response is we call an impulse response. And we actually have a very nice model called an impulse model that models this with two segments. And for an induced gene, and really a repressed gene would look just like the mirror image. For an induced gene, it will have some beginning level that you can call H0. And then it will, at some time point, it will onset, and then it will have a certain slope. And then at some time point, it will saturate and it will stay at this level. And then at some time point, it will offset, and it will go to a final level that can be the same, higher or lower than the initial response. This is extremely characteristic for all of these responses where you take a cell and do something to it that represents an environmental signal. In yeast, it can be something like this, right? You put heat shock on it, or you put hydrogen peroxide on it. In a mammalian cell, it can be something like it encountered a pathogen. It will have exactly the same temporal characteristics: bacteria, yeast, plasmodium, mammals. When cells differentiate, they're going to have a different temporal pattern. But this is classical for these types of responses. Okay, oscillations happen in a different set of biological conditions. They would happen in things like the cell cycle, the circadian rhythm, sometimes in yeast in something called the metabolic cycle. That's a very different set of situations.
I love the discussion.
I have no problem
and I
with
encourage
staying on this slide for the whole talk.
But we are, in the past, we've sometimes gone on for a very long time. So it really depends how many hours you want to sit there, the density of questions you ask. So do ask questions, but let's ask questions about the talk mostly.
I don't mind.
I know you don't. I want to hear the rest of the talk.
Yeah.
Okay. So nevertheless, with all the caveats that we have already introduced, we are optimists, so we wanted to do something useful. And what we thought was, let's try and think of the simplest models that we can for the regulation of gene expression. And we're still not very mechanistic, so for those of you who know biology well, this is going to be quite far away from what we actually know is happening in the cell before we even start the modeling. And that's very important, actually. Abstracting away most of the details tends to be a very useful thing to do. So we said, well, we want to model this as a probabilistic system, where we can model the expression level of a gene as a probabilistic function of what would be its regulators. And we don't care if the regulators regulate it in one way or in a different way, but because all we have is expression data, we're going to be assuming, and it's going to be a big assumption, that we can predict the expression level of a gene from the expression level of its regulators. And we do this even though we know up front that it is not the RNA molecules of the regulators that are regulating the RNA of another gene. There are all of these steps in between, and they might not be correlated at all. But we didn't have any of the others, so we worked with what we had. And we formulated it within the concept of conditional probability. So we said, what would be the probability function, density function, for the expression level of a gene that we represent by this random variable x, conditional on the level of expression of its regulators, which for now we're discretizing. They're not going to stay discrete for long, just for the cartoon in this slide. We're discretizing, say, the level of one regulator went up and the level of the other regulator went down, and we might represent this, this probability function as a Gaussian. Okay? So in this case, what we're saying is that conditional that this regulator went up and this regulator went down, this is going to be somewhat shifted away from zero, so maybe the gene is being induced. And I don't want to belabor this because this is not only older work, but it's the kind of work that we no longer do because we're so well aware of all of its limitations. But you can take all of this stuff and you can combine it together and you can try to learn based on this idea a Bayesian network. And this is a particular Bayesian network. It was learned from data that looked a little bit different than the one I already showed you. In this case, it's after deleting all sorts of genes in yeast, and it was built on 600 genes, and that was in itself a major endeavor algorithmically, just to get something that would learn and would finish in a reasonable amount of time. And the only reason I put this slide is that this is, again, one of those problems in trying to learn models in biology. This looks like a spaghetti. It is a spaghetti. There's a version of it that only identifies high confidence edges in it, in which case it becomes like a long list of little tidbits. And the bottom line is that even though you developed a major, right, you've put in a lot of investment, you used your best computational skills, it was the best data available at the time, although this is an old study, you end up with something that's sort of not really useful. And the best you can do with it is actually point to the things that you already knew and say, ha, we found one of those known things. Yes?
So when I see diagrams like this, one question I always have is, there are two uses of it. One is to use it in the machine learning framework where you're really just interested in predicting the response to a novel input. And there you don't really care what's there inside the network. But the second use could be that actually I'm going to take the network seriously. If I were going to do that, then you need uncertainties. In some sense, you want to define an uncertainty of your estimate of this network.
Right.
And that, some parts of the network you know well and some parts you don't.
And we did that, actually. So
You
we
better
are
repeat the
interested in
question,
the...
You better repeat the question.
Yes. Or next time, speak up. The second time that I'm repeating your question. He said there are two ways that you can think of a model like this. One of them is about how well does the model basically predict the data, how well does it fit the data, and if I try to generalize it to unseen data, I would provide it some level of expression of regulators, I would predict expression of genes, how well would I predict? And that's very interesting from a machine learning perspective, but you are absolutely right that this is not what we were after. We were after the topology of this thing, and understanding also the functional connections here, functional in a mathematical sense, so that we actually would get some mechanism out of it. Now, in order to do that, and that was the second part of the question, you need some estimate of the uncertainty in each of these edges, or even better than that, in other more higher order structural features here that could consist of, say, little tidbits of the networks with particular connectivities between them. And we actually did both of these things. We've done an estimation of individual edges and more higher order features, and we've initially done it with a bootstrapping approach, where you basically learn the model from slightly perturbed and missing the data in many different times, and you try to identify features that repeat across many different learnings, and from this you would call them high confidence features. And the other thing that you can do, you can actually cast this within a full MCMC framework and get an even better and more rigorous estimation of the features. The bottom line is that you end up with tidbits of features that were significant, but it's not clear how much insight you can learn from it. However, it wasn't an entirely wasted endeavor, besides publishing papers and getting theses and all sorts of important other things that happened along the way. It wasn't an entirely wasted endeavor because exactly because of this feature selection, it made us look at some of the structures that seemed to be the most high confidence in this. One of the structures that repeated itself the most looked something like this. So this is what we call a hub. People are familiar with this term. It would consist typically of a regulator, just in the sense of in the structure of this network. This is a parent of this children. And this single hub node would have multiple children nodes. Now, when we looked at it from a biological point of view, we realized that this gene we actually knew something about. It is what we call a MAP kinase, which means a regulator, although not a transcription factor, but a regulator of what is the cell wall integrity pathway. It's the pathway that maintains the cell wall of the yeast. And these genes down here are all known to be part of the cell wall integrity pathway. So we did get, and not just once, but in multiple instances, these little structures within the spaghetti that had what seemed to be the right regulator with a bunch of the right targets. So that was somewhat comforting. But the really important thing about it was not the comfort in the results. The important thing about it was that from this we realized that we were going after the problem. We were trying to do too much, whereas we could do less and do a lot better. And why? Because in the original learning of the model, we tried to learn each of these edges independently, right? And that meant a whole set of parameters for each of the edges, plus learning the structure. But if in the cell, and we actually knew that it was so in the cell, one regulator typically has many targets, and it's probably going to do something similar to them. Remember this image with the big green and red patches? All of these things were going up and down together, and they might have been regulated by one or several regulators that were shared across them, okay? So instead of learning 4 regulation functions with all their parameters, we could just learn one common regulation function, and this would give us 2 things. It was going to give us robustness, because these programs are going to be estimated from, we're going to estimate less edges with less parameters for more samples. And it's also going to be a lot more biologically interpretable, because we won't get a spaghetti. We'll get something a lot more organized, because the genes are going to be organized into these regulatory modules, okay? So as regulatory modules, we're going to call a set of co-expressed genes with a shared regulation program. And this led us to suggest a model that we call module networks, which underneath it, for those of you who come from machine learning, it is actually a Bayesian network, but it's a Bayesian network with certain constraints. And it's going to be motivated by the following biological story. Our story is going to begin with a gene. This is the gene. This is the region upstream of the gene that has sequences that are important for regulating its level. And this is the transcript level. And in some condition that we will call context A, the gene is expressed at what we will call a basal level. It means there are no special factor acting here, there's some RNA made. It's just a cartoon. Then in another condition, okay, the gene will be regulated by an activator. There's a protein, it's going to bind this, and the level of expression of gene is going to go up, and we mark this in red. And then in yet another condition, there's going to also be another protein around called the repressor. And when you have both this guy and this guy at the promoter, you're actually going to go below the basal level. Now, you can draw many cartoons like this. The question is, can we generalize this into something that we can also learn? So this is what the model is going to look like. At the bottom here, we're going to look at the target gene expression. But instead of just looking at one gene, we're going to look at a bunch of genes, because we are after these regulatory modules. And we're going to assume, for a first level of approximation, that they're all behaving the same and can be described by the same distribution. So there's an activator. The first question that we'll ask is going to try to discriminate between context A and the other 2 contexts. And it's going to ask a question on the activator. Is the activator induced? Because we're assuming that expression mimics the level of this protein. And if the answer is no, then we're obviously in context A, right? And we can model the expression of the genes, we'll do it with a Gaussian, to be unchanged. Then the other possibility, of course, is that the activator was induced, and then we need to ask a second question on the repressor. And if the answer is no, then we're obviously in context B, and the level of expression of the genes is going to be high. And if the answer is yes, we have a repressor, then we're in context C, and the level of expression of the genes is going to be down. So we have a regression tree here at the bottom. At the leaves, we have Gaussians that represent the level of expression of all of these genes in the module across all of the experiments within the same context. We're going to call all of this a regulatory module. This is what we call the regulation program, and this is what we call the module genes.
I'm sorry.
Yes.
I'm a little confused about why things are probabilistic. The data you were showing us, as was discussed, represents a mean of a population of thousands, millions of cells. You get one number. If you got that number wrong, which is a mean over a million samples, the probability is really low, right? I mean, you can't.
You get the numbers somewhat wrong all the time.
Right. So I'm not sure I understand what role the probabilistic character of the model is playing in actually fitting the data, which consists only of means.
The data consists of means across the population of cells that you measure. Absolutely. But if you repeat that experiment many times, there will be quite a distribution in the level of expression of genes between measurements. And I'm saying that not just by waving my hands, but because that type of experiment has actually been done. So for example, in yeast, there's a famous study that was published, I think around 2000, that did the same experiment 67 times, okay? And they measured, and these were just, isogenic yeast cells grown to meet log, and RNA taken out and hybridized to arrays. And there was substantial variation, and the variation, part of it was probably just measurement noise, and part of it was quite likely because it happened in the same gene over and over again in groups that seemed to make a lot of sense. Part of it was probably the fact that it's very hard to repeat the biological experiment itself in exactly the same way every single time. So if you just, it's not always exactly the same log, and they were a little bit happier with the hands of one technician and a little bit unhappier with the hands of somebody else. Maybe they spent a little more time before the RNA was taken out. The bottom line is that there's quite a lot of variation, and you can actually estimate gene-specific noise that is mostly probably technical noise. Unlike the gene-specific noise that we know exists and is intrinsic noise within the biological system at the level of single cell, that's a totally separate story. It's unestimable from particularly this. So in practice, the data is noisy. And sometimes you can, with the right and very cautious experimental design, really deconvolve all of the sources of the noise and where they come from. But in the vast majority of cases, people actually don't put in the investment at all at doing it this way. So you just have to handle it after the fact. So even though these are needs, and so that's one thing. Now the other thing is that this is an aggregation of multiple different conditions. They're not just replicates of the same thing. These, individual columns. And this is an aggregation of multiple genes that are not all exactly the same. But we want them to be grouped together because we want the benefit of not having to fit a regulation model per gene, which we realize is just not fitable from this amount of data. And we also want the benefit of taking multiple biological conditions and grouping them together within a context, because again, it helps with biological interpretability. So that's another level at which you would have variation, even if your measurements were perfectly clean, which they're not. There's another, yeah.
Just so I understand the free parameters in this model, the noise strength is a free parameter, the centroids of all the Gaussians are free parameters, the structure of this graph is a free parameter.
Yeah, you're going to see how.
So how do you know when to stop adding edges to your graph? How do you know when to stop fitting your data?
Okay, so we're going to learn it with an EM, and I'll show you, and you're going to reach a local optimum, not a global one by any measure, and you're going to be sensitive to the starting point. So that's just so to be clear, and I'll show you in a second how we actually learn the model. Then you also put in all sorts of things to constrain how would you put edges, for how long, and so on. For example, and they depend on the design of the data and your sort of biological wishes from the model in the end. So, for example, very often, if you know a little bit about how the experiment was designed, you might not want leaf contexts that would have 1 or 2 experiments. It might be too few. You might know that you even have more replicates per condition than 1 or 2. So you wouldn't want to allow it to split too much. And then you can put a certain error threshold, right, on how well can you fit or predict the expression at the leaves from the elaborateness of the regulation program, and once you reach that threshold, you will not allow it to continue to fit. I'll show in a second how we actually learn the program, and then I will repeat that point again, because it will be a little bit clearer in the context. But these are just so decisions. They're not very principled. The EM itself will converge, because it's an EM. Yeah?
So I'm a bit confused about your module, but you have one activator,
yeah,
and you have correlated genes.
Yeah.
Now, that's assuming too much on the causality. For example, I could argue there is an activator, and there are lots of genes that just happen to express because there is a downstream process, for example.
And you are absolutely right. There are a lot of assumptions that go into these things, but as I said, we were optimists. And we said, let's try it out, see what we get, and then see what needs to be improved. And I will show you at the end of the first half, if I ever get to the second half, I will show you at the end of the first half those cases where this works really well and those cases where it fails. And one of the simplest places where it fails, and it fails a lot, is exactly what you alluded to, that there will be a whole bunch of genes that are similar to each other, and the regulator will be relevant to some of them or not to others, and often actually, where things are just co-regulated really well, one of them will be picked to be the regulator, and it really isn't. It's a target just like everybody else. Now, one of the things that you need to do in order to make these models work better is that you need data that gives you enough distinctions, that there's something, that there is enough variation there to go around to actually distinguish things that otherwise would look indistinguishable from each other. And when we get to the second half, where we actually design the experiments ourselves, we could do a lot better than when we just had to use data that was published, which is this particular case. I'm moving to slide 4. Okay. So our assumption is that we can predict the target expression based on the expression of the regulators. It's a big assumption. We'll revisit it. We'll look at it more carefully later, but we just assume. How do we learn the model? Well, it's going to be structured in the following way. There are going to be two inputs to this data. The first is going to be expression data. This is just an image of an old array. This is the actual physical element from which those red and green colors are made. And then the other thing that we have as input is what we call candidate regulators. And this is where you put in, your biological hat on. And you say, and it's really based on the wishes of what you want to get out of your model. If you really want to have transcription factors associated with targets, then you need to put transcription factors as your candidate regulators. If you want to have signaling molecules as well, you can put them there. It's your choice. If you want to have metabolites and you measure metabolite levels, you can put them there. If you want RNAs, you can put them there. And the reason that I say that is that actually in subsequent years and in different permutations of this and all sorts of further development of the basic concept, both us and other colleagues other of my colleagues that have used this method have used many, many, many different things. So it's very flexible in this point. But it's going to give you, of course, as with all modeling-based approaches, what you allow it to be. And then the algorithm is going to be a structural EM that's going to iterate between two different parts. It's going to be initiated with a particular partitioning of the genes into modules. We use clustering, a particular type of clustering, but that doesn't really matter. You use some way of partitioning the genes. And then you're going to learn a regulation program for each module. And then you're going to take these modules, and you're going to forget about the assignment of the genes. And you're going to reassign the genes to the regulatory programs that predict them best. And then you're going to have a new partitioning, and you're going to iterate between these steps until you will converge. And the reason that you will converge is that this is formulated in such a way that the EM is proper and it works. And it's a local optimum. I'm going to go through each of these steps. This was just the overview.
Sorry, but what does EM say?
Expectation maximization algorithm, which is a common algorithm in statistics that when you formulate it properly will lead you to a local optimum, and you have a set of hidden variables and you're trying to estimate these hidden variables iteratively. So let's assume that we start with the partitioning and we're just looking at one module. So this is the module of genes as it came off of some clustering algorithm, and this is actually real data. Okay? So it's a group of genes, and you can see that in some conditions they go up, and they're red, and in some conditions they go down, and they're green. In some conditions they're not really changing. And there's a little bit more red than green, so the Gaussian representing all of these data points is somewhat shifted to the right. Okay? And our goal is to find one regulator at first, where if I split based on the expression of that regulator, it will give me the best prediction for the behavior of the module genes. And the way we do it is that we go through all of the regulators in the candidate list, one by one, and say we ask this guy, HAP4, when it goes up, and there's a split here, when it is not up, and there's a split here. Now, I draw this discretely, but you can actually set this threshold completely dynamically. This is not discrete in the real implementation. And then when it goes up, we can see that the module genes are actually going up very nicely. So now this Gaussian is going to be a lot more peaked and narrow and further shifted to the right. This guy is still, half green and half red. But I'm going to have a pretty good prediction. If I know something on the expression level of HAP4, I'm going to have a pretty good prediction on the level of expression of the genes here. Yeah?
So if I were to think of these as, let's say, vectors in Euclidean space or something, then the regulator has maximal overlap in some sense with all the other ones.
Yes. In some subset of the conditions.
Yes.
And that's actually very important. That's going to be a subset. Because very often what happens in biology is that the regulation is very contextual. This A will regulate B, C, and D in a subset of the conditions, but will have nothing to do with it in others. It's inactive and very inactive. It's just not relevant.
But if I thought of your module as a cluster in gene space, the regulator is like a centroid of that cluster in some ways.
Yes, if it was correlated throughout. But if it's just within a context, then it's only in a subspace.
The opposite, but you would say if the oppressor is going to be exactly.
Yes, then it's going to be anti-correlated. Yes, but then it's going to be
anti-correlated.
Then that you can switch. But the contextuality is the more critical one. It's the fact that we're only going to be in some of the dimensions and not in the others. So it's not just the context. So that's simple. And actually, if you compare how this performs, you'll see. I'm getting ahead of this. Now we're going to look at, another option, which is sig4, and we again try to find the best splitting point, and this is the best one that you can get. And you can see that you don't gain much information on the expression levels of the module genes by choosing this one. And you try them like this, one after the other, and you take the one that scored the best, in this case, Haskell. Okay, that's not the end of learning the regulation program. That's just going to be that top node at the top of the regression tree. Now we are going to continue to split on each of the subtrees. Say you try and look at this subtree, and there's actually obviously structure here in this cartoon example. You just can't explain it in half form. But if you ask a second question on this guy and the SEN4, you're actually ending up splitting it very nicely with a very peaked down regulation on this split and a pretty nice induction on this split. And maybe you could further refine it here. Now, this you're going to continue doing as long as the score improves, okay? Which is basically the fit of the data to the model. And how much do you want to continue improving, right? That epsilon of error that you still have is just a parameter that you set. It's not learned by the algorithm in any way. Okay, that was in response to that question.
Strictly, it's not as long as the score improves, it's as
long as the score improves up to an epsilon that you define.
Thank you.
Yes, absolutely. Now you've learned the regulation program, right? You have these modules, you have sets of genes associated with a set of regulators in these trees. Now we're going to reassign the genes to the modules. And how will that happen? So suppose we started with this cartoon example. There are 3 modules here, 3 genes, and 2 genes. This one is regulated by 2 regulators. This is just by 1 and by 1. And now we're basically going to take one of these genes, gene C. We're going to have to reorder the vector because the split is going to be different here. And we're going up, and we're going to test it also versus here. And we're going to see that the best explanation of the expression of this gene is given by this regulation program. So we're going to move it from here to here. We actually have to relearn the parameters here. Okay? We're not changing the structure, but by adding this guy here, all of these Gaussians are changing. So we have to redo them as well. And once we do that, we're going to continue to the next gene. And say in this case, we're going to test this one, and so on. And we go like this in a round robin for all the genes again, until there's nothing better to optimize, and then we stop. Okay? Yes?
You have to input the regulators. Can you learn the regulators also?
You learn which regulators are chosen from a set of candidates. The set of candidates is big, but you learn, some of them are chosen by the algorithm to be here. You could, and there's no limitation in the algorithm with everybody being a potential candidate. The point is that later on, the experiments that a biologist would want to do with this will invariably, they will want to have something like a transcription factor to see if it binds the promoter, or a signal transduction molecule to see if they can bring a small molecule and perturb it. It's not because it just reflects our experimental biases. Because frankly, if you change a major metabolic enzyme in the cell senses it. And it has like all of these cascades, and they lead in the nucleus, and they change gene expression. So you can say it's a regulator, and it is. But people have, traditionally felt uncomfortable about that. So your candidates is where you bring in your biased view of the biological world, and your particular ambitions and wishes. So this is how you reassign the genes into the module. And once you finish doing this reassignment, you forget all about those precious regulation programs, and you start learning those from scratch. And you might learn this one here with this guy. This entails some switching of the patterns, and this one here, and this one here. And it might end up being a different set of regulators than the set that you had individually. And you will iterate this process back and forth until you will converge. And when you will converge, you will have a model. Okay? So that's the, yes?
So although the network might have loops.
Oh, I forgot to, no. The network may not have loops, and I forgot to say that. Underneath this is a Bayesian network. It's basically a Bayesian network where you constrain a set of nodes in the network to share a set of parents. And because Bayesian networks cannot be cyclic, and because cyclicity cannot be explained well, right, within this probabilistic framework, then you cannot allow circles. And actually, every time you assign a regulator, you need to test that this does not impose a cycle of any length whatsoever within the system. Because, right, this regulator itself might be a member of this module. or of that module. And it can have all of these large indirect cycles, and that is not allowed. And the algorithm actually tests for that. And that's part of the reason that you have to do the reassignment in round robin, because that's the only way of efficiently testing for the lack of cycles. And I forgot to say that, and thank you for it. So when you do that, and we try to use exactly that stress data set that Audrey Gash generated in Stanford several years ago, we also did this work several years ago, there were 173 array experiments here, so there are 173 columns here. And now this is not like the matrix we saw before. So each patch like this is one module, and you can see that they're really pretty nicely tied together, okay? But the experiments are not sorted in the same way in the different patches, okay? Unlike in the big matrix we saw in the beginning, here, you know, this very rightmost column might be one experiment for this module and a different experiment for this module, because things got resorted based on how the regulators split them. So it's a nice bird's eye view, but it's one of those little confusing things that one has to be aware of. And to make sure that things are not too confusing, we're just going to look first at one module, which is this one that I highlighted in yellow. And here it is zoomed out, and this is just the result as the algorithm gave it to us. There is a regulatory program, which is this decision or regression tree at the top. There is the module genes here at the bottom. You can see that in some contexts things look very tight together, although there are always, right, these little blips of blackness within the red in response to a question on the noise. And there are some contexts that look noisier than others would. And that's quite typical.
You said regression tree, so this is like CART.
So this is like?
CART, the classification and regression tree algorithms.
It's similar.
It's similar.
It's similar in spirit, I would say.
Okay.
So now, and by the way, we'll get, if we ever get to the second half of the talk, using a tree here is just one choice. By now, people have done a lot of, including us, have done a lot of things that are not trees out there. And as long as you formulate the model correctly, and you will ensure that your EM will converge, it's perfectly legitimate, and we feel that it is within the same family of models. Okay, but the first one we did was like that. All sorts of people are.
So each gene is assigned to a single module?
A single module. It's a partition.
Okay, and then if you, supposedly you said that different contexts can be very different.
That's actually a partition.
If you had 2,000 conditions, you expect it to, at some point as you get more and more conditions, you expect this assumption to be less and less valid?
Absolutely. You're absolutely right. It's a partition because it was convenient. And it is not a good choice when your data set becomes very heterogeneous. Because what we know about these modules is that a bunch of genes might be members of a single module and be strongly, tightly correlated, even very close in Euclidean distance to each other. And then in a different set of conditions, that would break apart. Maybe completely, each of them goes with a different bunch of friends, maybe just with two pieces. There are all sorts of stories on all sorts of things in all sorts of cells. But this assumption of partitioning is just something that has been very convenient. It made the problem a lot more tractable. To solve it within the context of what we call biclusters instead of clusters, where it's only a subset of genes in a subset of conditions, and these both can change all the time, it becomes hard. And then the hardest of all is when you allow the same measurement to actually belong to more than one grouping. Because it has been shown that in some cases that actually is very biologically interpretable to think of things like that. But it's very hard. It's not hard to do this type of biclustering from the biclustering perspective, but from associating it with gene regulation, it has proven to be a hard problem. A difficult problem, I shouldn't say hard.
We should probably stop the formal lecture about within 15 minutes or so.
So we will definitely only be talking on these today. So how do we evaluate one of these features? Well, the first thing that we want to do is to make sure that the partitioning worked well. So for this, we want to test that functionally, the genes that appear in one module actually make some functional sense. Now, there are things called functional annotations, which are basically, labels, say oxidative phosphorylation or mitochondrion, which is a part of a cell. And these labels are tagged to different genes in the yeast genome, in our genome, by nice people who make beautiful databases. It's a very important endeavor. But once you have them, you can basically test for enrichment with standard statistical tests like hypergeometric, using, say, the hypergeometric distribution, and show that this group of genes is enriched from some functions. And this particular group of genes is enriched through functions like aerobic respiration. This is a particular way in which cells can make energy using oxygen and glucose in their mitochondria. So that seemed to be good. Now, the second thing we wanted to check is that the regulator that we chose, say this top one, the one that was chosen first and foremost, HAP4, actually makes sense in terms of regulating these targets. And it turns out that HAP4 is actually known to be the regulator of oxidative phosphorylation and aerobic respiration. And it's not just that it's known to regulate these guys, it's actually known to regulate many of these individual genes. So that was, nice. If it weren't nice, I wouldn't have chosen this example. Now, sometimes you might not have information directly that somebody knows it regulates, but these DNA binding proteins like HAP4 or this guy here, MSN4, they actually bind pretty specific sequences. We call them motifs because they have varying affinities to different variants of these sequences. Okay, so we can again represent them statistically, and we can test within the promoters of these genes if there are the relevant binding sequences for these factors. And we find that both the binding site for HAP4, which is this sequence here, and the binding site for this protein, MSN4, which is this sequence here, are enriched in these genes promoters compared to the genome background. So again, so it makes sense that these are true regulators for this system. And then finally, you want to make certain that there's sort of a match between what this program says, at least about the logic of gene regulation, and the condition that we know actually is true. So this program actually says when HAP4 is induced, the genes in this module will be induced, and it's not shown here in words, but it actually says that this happens in things like stationary phase, which is when you take glucose, which is when you roughly take glucose away. This is actually true. HAP4 is an activator, and it induces the expression of these genes, and we know that that happens when they enter this phase called stationary phase. So this is sort of, the best of everything. Everything worked really well for this module. Now there are 50 modules. How did we do across all of them? Well, the modules themselves were coherent based on annotations in 41 of 50. We found a match between regulators and targets in 30 of 50 modules. We found a match between regulators and cis-regulatory motifs in 15 of 50. Now, this is an old study, and we knew very little about cis-regulatory motifs when this work was done. This number can probably be improved today. And then we found a match between the regulator and the condition and logic in 35 of the 50. So we felt that there was something that made so few assumptions and was so far off from what really happens in biology, right? It was all just from expression data, no other information used. That was, pretty decent. And then we said, well, can we test a little bit these predictions that we made? So I want to go through these slides quickly, mostly because I want to get at least to the justification of why this works at all. But we said, this looks nice, but there are also a lot of things here that we couldn't say that they mismatch. We just couldn't say anything about them at all. The regulator was not characterized. We just knew it was looking like a transcription factor based on its sequence, but nobody knew what it regulates. Or it looks like a kinase, a protein that phosphorylates other proteins, and these often have a lot of regulatory functions, but that was basically all that was known on something. So we said, for this we need to characterize as best as possible the function of those regulators in the model, and we wanted to do it across the whole model. So in the columns here, we see the different modules, right? And I already showed you that we can test which binding motifs are enriched in them and which functional annotation. Now each module, as you remember, is associated with a set of regulators, right? And now if we take these two matrices and we multiply them, we end up with an annotation of the regulators. So if a regulator was associated with more than one module, and both of these modules had different or the same annotations, this information will aggregate in this matrix. Now we can also take these splits, right, between the different contexts, and we can label the different biological conditions, and we can test for an enrichment on splitting different conditions away from one another based on the split that the regulator induces, and from this we can get a matrix of regulators by conditions. Okay? And now when we actually do it in real life, this is what we get. Here are the regulators by motifs, annotations, and conditions. Here is a zoom in for one particular regulator. It's called GAT1. The reason it's called GAT is that it binds a site called GATA. So that we got right, but we also associated it with three other things that come along for the ride because the same genes are regulated by multiple things. We know that it regulates these functions, amino acid metabolism, sulfur metabolism, but we also got these guys, ion transport, cell wall, that we didn't know is associated with. And we know that it acted in nitrogen depletion, amino acid starvation, and stationary phase. So it was a pretty good one. This is one of those that we would consider well validated. But then, of course, there were many other things that were, so it's a comprehensive, although somewhat noisy annotation. But then there were other things, like these 3 columns here, for which we actually knew, very little. Actually, we knew almost nothing about them. This one didn't even have a proper gene name at the time. These ones have gene names, but there was nothing known about what they actually do in a yeast cell. And we had pretty specific predictions. We said, this is a kinase, it won't bind DNA directly, but it would act through this binding site. And it would affect glycogen metabolism, and it would be acting in vitro. And we said this transcription factor should affect protein folding genes in stationary phase. And this thing, which is a phosphatase, should affect these processes. They're essential for actually making genes, making proteins. And it would act in hyposmotic shock. Okay? These were very focused predictions. And that was the important thing about them. Not that they were correct, but that they were actually testable. Because what happens very often with genomic studies is that they're beautiful and they're big, but there's nothing specific to do after to tell if they're right.
The color is...
Oh, the intensity...
10 minus 4, so...
Yeah, the intensity for the condition, anything that's less than 10 to the minus 4 is going to be the maximum brown that is shown here. This is just a cartoon. The real colors are here.
And it's also relative? It's also relative. It's relative intensity.
This one, yeah. So this one goes from white to maximal color on each of the scales. So before it was blending 2 colors, this one, or there were 2 scales actually that met at the 0. This one, because it's for percentage or for p-value or minus log p-value, then it goes just from white to maximum intensity. Sorry, I forget sometimes to get used to a certain visual representation. It seems obvious, whereas it is totally not. And by the way, you can fool people tremendously with these things, with how you choose your intensities and where you want to saturate them, and people do that all the time. Very bad habit. Anyway, so this made a very focused prediction that the regulator X, these would be one of the X's, will activate some process, Y, these would be these blue things, under certain conditions, Z. And that's something that you can biologically test. So this brings us back to our scheme, right? We observed expression data, we modeled them, and now we made specific predictions that we can actually test by perturbations. And the way that you perturb, and this is the pleasure of working in yeast, yeast has what we call very good genetics, means that you can go and you can zip out a sequence out of its genome and put something else there that's inert and just lets you select that you did what you wanted to do, and the gene is gone. We call it knockout. You can do many other beautiful genetic tricks with yeast, which is why people love them and want to work on them all the time. So you knock out X, you just take it out of the genome, then you put them under condition Z that the model predicted, and you see what happened, and you're going to have three conditions for saying that something was successful. You're going to ask whether there was actually an effect under the predicted conditions when you knock out the knockout. The reason that the genes have no annotation is that when you actually knock them out in just good growth conditions and no stress, nothing's happening to the cell. So you need a guess at least of what to do to the cell to make them do something. And then you would look whether the response happened in the predicted modules, and then you would also look at whether genes from the predicted processes were responding. And you can test for this with a paired t-test, look for differentially expressed genes. You can rank modules or do enrichments for differentially expressed genes. You do also, these are just bioinformatic little things. You can test whether the regulator process annotations from the matrices are enriched in differentially expressed genes. The really important thing to know is that we got all 3 regulators validated. And the really cool thing that I'm still happy about many years after this study was done was that we actually collaborated with the biologist lab, with David Botstein's lab, and there was a great postdoc there, Michael Shapiro, who actually ran the experiment. And he was convinced that we should test these under different conditions. And we argued for like a month what should be done, and in the end, David said you just do both experiments. You do it in this condition and in that condition, and the one predicted by the model worked, and the one predicted by Michael did not. And he shouldn't be bloated. He's a really good guy. Okay, so how many minutes do I have left?
Another 5.
Okay, so we'll just finish today with yeast. I'll just zoom into one slide to show you that this actually works in animals. Why does this work? This should have failed miserably. Okay? We made a big assumption. We said that the expression level of a regulator is going to be a proxy for its activity. But we knew before we ever started this study that this is not correct in biology. Okay? What actually controls the activity of a regulator is several steps removed from having its RNA in the cell. If there's no RNA, there's not going to be any protein, and hence, obviously, there's not going to be any activity. But the fact that you have the RNA is many steps away from having something that's doing its job. So here's a little cartoon example. This is the guy, SLT2, that the whole, endeavor started with. What happens when cells are put under low osmotic pressure is that SLT2 becomes active, it phosphorylates this guy, it's called RLM1, it binds the promoters of genes, like this guy, CRH1, and it's going to make RNA. Okay? And if you measure it on a microarray, you're going to see your red signal. The reason this works is that RLM1 doesn't only bind the CRH1 promoter, it also binds the SLT2 promoter. And SLT2 too goes up as well. And this is classical positive feedback, which is extremely common in these types of responses. Okay? Many regulators are themselves transcriptionally regulated by the systems that they control. It can be autoregulation, which we could draw as this, right? They just bind their own promoter and activate their own expression. It could be negative, it could be positive or negative. It could be feed forward, which means a regulator activates another regulator that then activates expression of a whole set of genes. Okay, so we would be able to identify this guy, we would be able to identify this guy. It can also be feedback, just like this one. Okay? Goes and feedback, yeah.
So there are MAP kinase phosphatases that would negate this.
Yes.
That can be taken into account.
They will also be, by the way, the phosphatases are one of the most transcriptionally regulated things out there. And they're actually part of a negative feedback loop in terms of the logic of the system. They're very easily identifiable, both in yeast and in mammals. There is work both from us and from other people showing it in several responses. They have a very particular temporal pattern. Their onset time in this impulse is very characteristic. They're a second wave of thing. You can pick them up like this and know what they're doing. So expression does give you signal. And the only reason that it works is that, the cell makes it work for us.
So this is slightly ironic, right? Because you have a model which explicitly neglects feedback,
yes.
to identify a forward
yes.
arrow, which you shouldn't be able to identify,
yes.
but only because there's a feedback arrow that goes
yes.
the other way.
Yes, and I think that's the beauty of the whole thing. Now, if you work with the time course data...
There are two views, right?
Yes. Well, it works, right? And if your goal is to really find the first draft by which you could go after experimentally, and I'll show this in a slide in 2 minutes or 1 minute. I don't know even how many I have left. But if your goal is really to find the circuitry, right, rather than building the ultimate algorithm, then all you need from the first pass algorithm is a good enough draft to identify the right candidates for experiments. You actually care extremely little on what we used to care a lot about, which is, do we get the edges correctly? Do they target the right modules? You really want the right players roughly at the right place under the right conditions. The rest can be handled, the next iteration should be handled experimentally. And with the next set of experiments, you could build a much better model. Then you could direct the next level of experiments. And you could start also identifying those things that are not transcriptionally regulated at all. Because then you won't necessarily be beholden to expression. But historically, in terms of the work that we've done, this is how we started. We knew that there was feedback. We ignored it egregiously, and then we ended up succeeding only because feedback was there. But cells are so much smarter than we ever will. Yes?
So what you're saying is essentially, it makes sense, that it's a hypothesis generating mechanism. Now, are you in your research program now teamed up with an experimental group so that you are generating hypotheses?
We're teamed with them very intimately. We simply became an experimental group to do that.
Okay.
Because I will tell you what. Because this was one type of work. There are many of these, different people have different ideas. They became cleverer and cleverer. The algorithms became more and more elaborate. And nobody had any idea if the algorithms were getting anything right. Because, this was one of the most heavily tested models out there. 3 regulators out of, 100 or so that were chosen by the model. that was like nothing. How could you ever improve the algorithm if you don't know if what he gave you is correct? You knew that it wasn't totally off, right? You had all the corroborating evidence from what was already measured, and that was used. You can't do that in mammals. There's not that much known. So it was obvious that we couldn't continue this way. And what you really need is to set up the right experimental system together with the right algorithmic approaches in order to really build those things.
So you're on your
It's time for the one mammalian slide.
Yes. So I'm going to skip this, and I'm going to show you, first of all, that we do this in a particular type of cell that has a lot of these nice properties that yeast have. And I'm going to skip there a while. Okay, this is the slide. Okay? And this is what we do, and we actually have done. So now again, you have these little things, but you're no longer stimulating with heat shock. You're actually stimulating with a much more specific molecular signal. This signal has a very specific receptor, and the signal is a component of a pathogen. Okay? So this guy senses gram-negative bacteria because it senses lipopolysaccharide, which is a component of gram-negative bacteria. This guy senses viruses because it senses the RNA of viruses that we can mimic synthetically with poly-IC. This guy senses gram-positive bacteria with something called PAM3CSK, which is a component of gram-positive bacteria, and so on. Okay? And then you profile their expression, exactly the same experiment as you did with the yeast, but with dendritic cells that you can isolate from your bloodstream or you can isolate from mice. This was done in mice, we also do the work with humans. And these cells, they're the sentinels of our immune system. If they go wrong, things go really, really wrong. You either are extremely sensitive to certain infections or you end up with an autoimmune disease. So they're important guys. Okay? Now we use exactly those types of models, but because this is a time course, and we spent several years playing with models, we actually use time in our benefit because now we can worry a little bit less about feedback because we're going to use lags in time between the regulator and its targets. And it tends to actually give you somewhat better performance, but then you get this bunch of candidate regulators basically, and conditions in which you want to test them. And then you're going to perturb every single one, not 1 or 2 or 3, but 100 or 200 or 300, because you can do that today with good perturbation reagents. And then instead of measuring all of this, which is super expensive, and not just costs a lot of money and time, but requires huge amounts of cells, which are very hard to get from either mice or people, because now you have all the perturbations and all the conditions and all the time points, too much. You're going to measure it only for a signature of genes. And there are new technologies that allow you to do that. They actually look like in cartoon form, like this. So you're going to perturb the regulator, stimulate the cells under the condition you want, the time point you want. You're going to see the effect. And then you can build a real model. And of course, that model will still be a genetic model. It's still not fully mechanistic. But now you can take this scheme, and we actually do that. Now you can take this scheme, and you can devise new assays to do on a very large scale, binding assays for transcription factors to promoters. And you can measure all of them in the native state, and you can measure them under perturbation. You can develop schemes to actually, or experimental methods to actually measure transcription rates. We do that in metabolic labeling. And when you do that, you can again couple it with perturbations, see the effect. So you can get a model better together. You can use phosphoproteomics today and standard proteomics today to actually see the effect on protein levels, and protein localization, and protein modification. So it becomes a very generalized thing. The output is still the same output because that's the output we actually care about. We want to understand what causes RNA levels in the cell to be what they are. And then when you have a really good model and you understand a lot about the players and the connections, you can go single cell, okay? And you can start asking questions about variation. You can go into genetics, and you can ask questions about variation in gene expression that are genetically encoded. Because if you take dendritic cells from different people, if you take dendritic cells from people who have lupus, they behave in really bizarre ways, okay? And if you take dendritic cells from mice or from mouse crosses that have all sorts of phenotypes, their expression looks very off, and in ways that explain why, they have high susceptibility to certain types of viruses. So there's a way of really wrapping this together, and for us it started, in the lowly yeast and in that model. We still use a descendant of that model, it's fancier, it has bells and whistles, but the basic idea is the same. But the main thing that we learned from doing that work is that work brought us here, right? And that the only way of getting to something that's refined and understandable and correct in biology is to constantly go back and forth, biology, experiment, algorithm. Plus, on the algorithm side, this actually opens very interesting questions in active learning, which we couldn't tackle at all before because there was no activity after the learning. But now we can. And then you can figure out how good they are. And they're actually really, really bad, the outcomes. So with that, I'll conclude. I'll show you the concluding slide of the dendritic cell so you get the numbers. So from just that scheme that I showed you in the beginning, there were 40 things that were all associated with regulation of gene expression in dendritic cells. We found 60 new that are validated now as real, and their connectivity. We found a whole co-opted set of cell cycle transcription factors that are controlling the antiviral response in these cells that are not cycling. We found 12 regulators that then came up in genome-wide association studies for immune disorders. We found how specificity is encoded. We found these chromatin-based mechanisms. And this is a very general strategy. We've since applied it to signaling molecules. We have a small molecule that we can give to a mouse and it loses its ability to find viruses completely. So you can really do it. It's not even very hard. It just has to be done in this very systematic way. And I want to finish by thanking the people who did the work. From my lab today, the person I should thank the most is Ido Amit, who is a spectacular postdoc. He's going to start his own lab at the Weizmann Institute later this year. And he's initiated all of this work in the dendritic cell. The work that I showed you on module networks was done several years ago. Starting to be embarrassing how long ago. All of these people who were grad students when we did the work now have their own, have had their own lab for a while now. So it was done in collaboration with Nir Friedman from the Hebrew University in Jerusalem, and Dana Pe'er, who was his graduate student and is now an assistant professor in Columbia. And it was done in collaboration with Daphne Koller, who is a machine learning professor in Stanford, and her graduate student Ann Segal, who is now a professor at the Weizmann Institute. And it was done in collaboration with David Botstein, who was in Stanford, and his postdoc at the time, Michael Shapiro. Thank you very much.
So we have time for three questions in this room, and then we'll go out there and everybody can have Aviv as long as she can stand it. So
yes.
So one of my biggest worry about Bayesian networks is usually that you see correlations and the model imposes a causality on it, right?
Yes.
Now, what I was wondering is, you showed a happy scenario where things worked out, but even within the model,
yeah, I owe you that.
If you have other networks, other hierarchical structures with a different causality, which are equally probable or close by, in this optimality.
It definitely fails a lot. It has 3, I think, major modes of failure. The first mode of failure, the regulator is regulating, it's not changing the expression, you don't see it. That's all. The second and most major mode of regulation, because it's a false positive, is the regulator that you choose is just being co-regulated with your target, but it wasn't your candidate set because it happens to be a transcription factor. And then you choose it because it looks awesome as a regulator, right? That's a major mode of failure. It usually ends up to be important for something, so if you perturbed it, you will learn what it was important for, so you didn't lose that much by going through it, but in terms of the model itself, it's a real mistake. And then the third type of mistake that you get is in feedback scenarios, you get often a mistake with the logic, because when you're in a negative feedback loop, the regulator is going to appear as a positive regulator because it's co-regulated with the target that it's actually indirectly going to suppress. You are nodding like when I said negative, so you realize that. So that's the third major mode of failure. There are all sorts of other subtleties that are there. And yes, these are not positives. Now, the one thing that does help substantially is when you have a time course. When you have a time course, you can use lags in time, in addition to using just basic, correspondence by correlation or whatever other measure of fit you have between the regulator and the target. And we have used those in dendritic cells because there we did the experiment and we did a very, very careful time course and we had many time points and the resolution was right. But I do have to say, when we originally designed the experiments, I had huge hopes and we put a lot of investment into the time course side and building a model, and it now uses this fancy regularized regression approach. It's like all bells and whistles, but the bottom line is that it still tends to fail in exactly the same places that it failed before. Maybe a little less so, but not a lot less so. And that's exactly why you really need to test the predictions of these models. You need to assume that they're getting a lot of the topology wrong. They're probably getting the layers and the processes that they regulate and the conditions in which they act reasonably fine. fine enough to invest in an experimental piece.
Are you saying the perturbation helps you correct the error?
The perturbation, yeah, because after you do the perturbations, you observe again the effect. And with that, you can relearn the model. And you can relearn the model, for example, to constrain a regulator to affect directly or indirectly in the model through a chain the target genes that are affected when you perturb it.
And so it detects the error.
So then it detects the error.
It doesn't detect the first error if the regulator
No, if the regulator is not...
Then
no. But that's why you need to measure other entities in the cell. So for example, once you identify regulators that are functional, you can go after, and I would have shown it had I had time, you can go after it with things like proteomics. And you can look for things where modification states have changed. And by this you can find a whole new bunch of regulators. We found a whole new branch for antiviral regulation that acts in all interferon responses, no matter what the stimulus is. And we started with a single. Out of all of the proteins that are active there, only one is transcriptionally regulated. And by the way, this is also a principle that we knew from yeast. So in the yeast cell cycle, where you have primarily a system that's driven not from transcription, although it affects transcription tremendously, there is beautiful work done by Per Bork, not by us, to show that very often when you have a whole system that's active and is cycling, one component will be transcriptionally cycling, and the rest of them will be flat. The same seems to be true for many other types of responses, including those in network cells.
I'm going to
So
take the privilege of the chair and claim that that was 3 questions. Aviv, the marvelous talk.